Lisa.
I don't know that Lisa's on yet.
I thought she was. Something happened? I don't see her actually, you're right.
Just wait another minute, maybe she'll come back on.
Did you start? I mean,
no, I was like, I think she dropped out and then we're waiting for you, so go right ahead.
Yes, gosh, my internet bleeped. Good morning, everybody, or good afternoon. I'm glad you all are here. I just wanted to tell you that we are going to have an in-person meeting at PAG. It's going to be on Monday morning from 9:30 to noon. We have asked for a room, so I'm sure we'll get one. If anybody has extra money and they want to pay for some audio visual, that would be very much appreciated. I think it's about $1,000. But at that meeting, we're going to talk about the future of ag biodata. We're going to talk about the grant we're writing. We're going to talk about a couple other things, and we really hope all of you can be there. It's a time at PAG where it's sort of a low, there's not another workshop going on or anything, so I'm really looking forward to seeing all of you that can attend there.
All right, and for those who can't attend, we are still trying to work on some, at least setting up a Zoom, a way for you to attend verbally the meeting, but it's still in works. I haven't got that confirmed yet, but we'll keep you updated. So for the short presentation today, the Ag Biodata Steering Committee invited the developers from BTI and BreedBase to come and talk. Their speaker is, oh, please do a good job. Gnome, how'd I do?
Guillaume, yeah, that's right. That's good.
All right, good. I had the pleasure of working with him earlier in March, and he gave a great presentation in March, and I'm looking forward to another great one today. Just a good one to find out what's available, and I'm looking forward to an interesting conversation to see if others can learn from BreedBase what they have available. So I'll hand it over to you. Thanks
Thank
again.
you, Jacqueline. Thanks so much. Hi, everyone. My name is Guillaume, and I work as a postdoc in Lukas Muellar's lab, where we've developed BreedBase, a database for breeding programs. And today I'll give you an overview of our system and how it links to other systems and how it can do for plant breeders and plant scientists. Just to give you a brief background information on the team, if you haven't met Lukas or anyone from our group before, Genomics has been busy and focused on developing bioinformatic resources for the Solanaceae community over the past 10 years. And among those resources, one of the major one is the development of the resource for the genomes and their availability online through the website Genomics. From that time, we have moved to several fields around genomics and breeding, including genomic selection or expression data analysis, for example. And today, I want to show you how Genomics has now moved to hosting resources for genomics in Solanaceae, of course, and several tools. I put you here a short summary of what's available on the website currently. So you have familiar tools such as BLAST and Expression Data Atlas, JBrowse, and so on, and a number of reference genome for various crops, and a lot of resequencing data. But also, we also have released more recently the Expression Atlas, where you can find several experiments being performed on tomato. Out of Genomics were developed in the last 5 years a set of other databases, which include databases on cassava, banana, sweet potato, or yam, and a few others. We have named these BreedBase as they are all using the same engine behind, but they are of course, adapt to the different crops and communities that are working with these. And today, I will give you a round trip of the functionalities of the system, which is, of course, focused on plant breeding. So here, to summarize shortly what you can expect, of course, breeding is a lot about cycling over and over on evaluating resources, analyzing data, selecting it, and performing subsequent crosses. The slides here are giving you an example through cassava, which is, of course, one of the main crop we get to work on these days. So next-gen cassava, there was a special attention given in the past years to develop predictive breeding using genomic selection. And for that purpose, was enabled in the cassava database, cassava base, a tool focused on on genomic selection we called SoilGS. This tool is mainly working on high-density data, including SNP data, and uses the phenotypic resources which are stored in the database. You can also check the publication related to SoilGS, which was released a few years back. From SoilGS, the use have been expanded because, of course, predictive breeding was in attention of the project, but also developing stronger resources in different ways for breeding were also required. Therefore, to achieve good genetic gains using predictive breeding, you also need good genetic data and good phenotypic data as well. And to perform that, the needs were various, including the harmonization of the data resources, and that goes usually through standardization of procedures. Those procedures have a lot to do with ontologies, for example, but also for various activities such as tissue sampling or data collection in general. As well, aggregating resources also mean that you have to work together with a large community. And for that aspect, the database has been instrumental as well. Talking about ontologies, I want to show you what the database currently does and how it handles it. And perhaps there will be space for discussion on that based on each other's experience. So this is a snapshot of the current ontology we use in cassava. It's developed together with the crop ontology team based in Montpellier. And it includes various set of traits, including agronomic, morphologic, physiological, quality traits, but also more recent types of traits such as farmer survey trait, remote sensing data from drones, for example, or from various tools for analysis of infrared spectra. From this point, the database is able to catch on aggregating data since the traits are available in our system. Without that, of course, it's hard to perform data collection using BreedBase. Here I give you an example of what can be a trait, as simple as this, with a 1 to 7 range for storage route, and with a qualitative observation. But it can also be quantitative using several types of scales, metric, kilograms, and so forth, of course. Further than that, developing the ontology being one, maintaining it and enriching it is another aspect that we have commitment with. To do that, we, of course, based our ontologies on crop ontology in initial developments using trait dictionaries, but the maintenance has been now a lot moved to other forms of file formats, such as OBO files. I guess some of you may be familiar with that, or OWL files. Several tools exist in the community, and we used to have OBO Edit as a curation tool that we have now moved to Protege to perform that task. Another aspect, which is of critical importance when working with a large community, is to collect requests from users. And for that purpose, the interface has to be designed for the type of user we are working with. Our users are usually breeders. Therefore, their interest is very practical, geared toward what they want to collect in the field. And for our developers and the folks who are curating the data, the interest is usually more in the bioinformatic aspect of it. Therefore, the platforms have to communicate properly. for that aspect, we developed this interface called submit.rtbbase.org, where users can submit requests that are then being transmitted to us through a GitHub platform from the Plantium project, and where we actually add new information to the existing files we maintain for each ontology, including OBO files. From this part, we can interact also with other colleagues and other websites for ontologies. Now, this said, ontologies are very practical to use for storing simple traits, but when it gets to more complex traits, which usually is needed, such as a trait that combines a method for a particular data collection on a phenotype, but which also require, say, a particular component, either chemical or a particular time approach, it becomes quickly difficult to aggregate all this information in an easy way for the user in a single ontology. So our approach was to do what we call post-composing, which is basically the aggregation of several sets of ontologies which are dedicated to different activities. And combining these, as I show here on this snapshot of the database of Cassava base, you can combine, for example, an ontology that would have to do with different methodologies to process cassava roots using particular morphological parts of the root and for a particular chemical component. Combining these would give you a composed trait that you can find in the search tool from the database. And that search trait can be parsed later on into the different components that it is made of. That's how far we are today in terms of ontology management. Now, for the time we have left, I will give you a broader overview of what the system is able to do, and in terms of its various components I showed earlier. So evaluation of resources, analysis, selection, and crosses. So the database includes different search, including what we call the wizard search, which is a multi-criteria search that allows you to perform multi-queries on breeding data, including location, years, trial experiment types, different germplasm resources, breeding programs, trial types, I think I said that, et cetera, et cetera. Beyond that, you can generate your own trial experiments, your own barcodes, and related resources such as tissue sampling for genotyping, the image collection, or the storage of genotypic data associated to the trials. Of course, there is a component to associate for the geolocation to each of these activities. And finally, and importantly, as I mentioned earlier, phenotypic analysis can be combined to genotypic analysis through SolGS, but also simpler tools such as summary statistics, selection indices, analysis of variance, and very soon a mixed modeling tool will be released together with a new GWAS tool. And finally, the pedigree cross activity can be overviewed through the pedigree viewer. I will show you a little further, as well as a new interface for cross management using different apps. We understand that there is a lot of activities in breeding, and therefore it can be difficult to navigate through a website when you're a breeder, and things can be complicated over the day between the field and the office. So we try to simplify our interface by developing workflows, each of them being designed for various activities that the breeders have to deal with on a frequent basis. That includes the design of an experiment, the access to seed resources, or the data collection for phenotypes, the performance of crosses in a crossing block, some analysis components, and so forth. So here I give you the snapshot of one of these workflows. This workflow is designed to upload genetic data into the database. It basically expects you to provide a VCF file, which is one of the most common formats for genomics resources, and bring it to the database, ensuring that these accessions have been previously uploaded to the database, or possibly if you have used tissue samples from the database, it would check that availability and force that information to be there before it actually uploads it, and ask you to provide a protocol regarding how the data was actually generated, and finally load it into the system. After showing you this set of main functionalities, another aspect I want to cover briefly is how do we use BreedBase when we are in the field? Well, we've been working tightly with Kansas State University and the group of the phenoApps, which have developed a set of applications that can be useful for various activities along the cycles. One of them is, of course, Fieldbook, you may have used or heard of. Another is Coordinate that we use for tissue sampling within the Next Gen Cassava project. And a few others include an inventory app, an app to verify barcodes and content. And finally, one more recent app that has got a lot of interest from breeders to perform crosses in the field. All those apps are relying on the barcodes that we generate from the database, so that there is a continuity between database to the app, from the app to the database, in terms of import-export of information. Here, an example of this application is the Fieldbook, using those barcodes and scanning the barcodes. It would assign to the field out the related plot you are actually scoring. I should have mentioned earlier, sorry, that we expect users to use the database to design or load the trial, so that you have your barcodes generated from it, assigned to the plots, and finally you can go for data collection using the Fieldbook, uploading back the data to the database. A similar approach has been taken for the tissue sampling using Coordinate, where the same workflow would apply, using the same barcodes assigned to the same plot, but this time you would rather link wells in a plate, for example, that you would send for a genotyping assay to any platform of your choice. Okay. After talking about the database structure, the apps we do use with it, I want to mention the breeding API, of which BreedBase is a large user. So if you're not familiar with the breeding API, it's a programming interface that was developed in the past 3 years to ensure exchange of information between different platforms, including BreedBase, and other tools developed by the breeding or genomic community, plant communities. It includes packages in R, in JavaScript, and is available online. I will provide references to that later. Its main strength is allowing the exchange of information in a standardized way, so that there is no loss or corruption of the information and a continuity between systems. Therefore, you can leverage from this environment to develop applications that can be used by various users and understand each other. Here, to exemplify it, I like to think that systems are different bricks, and BrAPI is really the cement that can aggregate them together. Knowing this is BrAPI.org, the website where you can consult to find more resources, either about the code or other community aspects, as well as a GitHub repo where you can find the R package using BrAPI. And this is an example of one of the community meetings that are occurring every year in various places around the globe. This year it was in the Netherlands. I think very soon it will be in India. From BrAPI, having BrAPI implemented in BreedBase allow the use of various applications that we like to call the BrAPs. And those BrAPs include the pedigree viewer I mentioned earlier, which is embedded in Cassava base, but also in other BreedBase instances and can be installed in other databases using BrAPI, as well as a more recent developed tool which has to do with field map viewing and geolocations using GPS coordinates. Genetic map viewer, also using BrAPI, of course, and which is able to focus, zoom in and out real time for multiple identity maps. And more focused on the breeding aspects, we have a tool to compare trial performance using scalar plots and histograms. We have a box plotter tool, the BrAPI app package I mentioned earlier that allows to pull data from any BrAPI compliant system and analyze it. And a graphical filtering tool which allows selections on a visual basis of multiple sets of traits over several experiments. Another aspect after those technical components I like to mention is how important for our communities is to meet and to meet together in the field, in the office, on a frequent basis to build on those technical developments that allow improvement of the routine breeding activities. That's where we can really improve the functionality of the tool we aim at developing for the breeders. Through the Cassava project, it's something we had the opportunity to do in several occasions and different countries. And I cannot underline enough how important that can be to make data development and aggregation efficient. And a good explanation or result of this is to see what is the content today of a database such as Cassava base. This may be a little old, June 2018, but you can see that we have a fairly high amount of information stored there in various ways. Of course, data quantity is one, quality is also at stake, and we use those opportunities to visit to improve quality management across the different programs, across our own team to really understand what are the key points we need to address. The last point of this presentation is maybe looking toward the future and where do we go with BreedBase and how can we sustain it? I guess it's an open question, but for elements of reflection, just want to mention we worked on BrAPI over the past years, but we are also now focused on image data analysis as infrared spectral analysis tools that can be included in the system. We have a lot of attention to genotypic data and how it can help breeders to make quality control. But I guess another aspect is collecting more environmental data in the future. Make sure the decisions that are done by breeders are fitting the environment they want to select for. And of course, an aspect is adoption of the system and making sure it's sustainable. In that regard, I just want to mention that recently we had the Breeding Insight project that has developed around BreedBase and the phenoApps using BrAPI. So that's always positive to see the tool gets attention from institutional partners. And with this, I'd like just to bring together a set of information you may like to check regarding the code, the applications, or any tutorials you may want to check. Of course, the slides will be available to you after the call. Thank you so much for attending. Thank you.
Thank you again. I'm going to unmute everybody, and you're welcome to ask questions. This was a great. I really like the ontology in the beginning of how you had that organized. Is that all manually done, or is that something that you have some sort of computer code do?
You mean structure the ontology, or?
Yeah, the main structure.
I'm afraid we had to do that manually. Well, I mean, half and half. The ontology, of course, requires a lot of curation from experts and bioinformaticians to organize it in a structured way using OBO files or AL files. But of course, it cannot be fully automatized, at least for the very early steps. It requires a lot of commitment from the community and the bioinformatics team to put together, sure.
Mm-hmm. How often do you have to go to Africa for various countries in Africa?
Well, I guess it depends of the projects and the level of how to say implication, the level of implication we have in these. But regarding cassava, it's an important project to us. We have a lot of partners there, which are both based in East and West Africa. So I would say maybe a couple of times a year or 3 times a year is a fair amount.
That's amazing. That's great.
It is.
I like the part about getting the community together, you know, as part of the project, working on the community.
Yes, absolutely. For us, it's been really a tremendous help because knowing about the traits is one thing, but knowing the actual challenges to collect them is another important one, so that we can address better the tools we develop and how we design them, make sure that they are matching the actual, you know, environmental settings people may have there.
Yeah.
Any other questions? Sorry, I haven't looked into. I know people can raise hands and all of that.
I have a question. Thanks for the talk. So if I understood correctly, BreedBase is now a merge or conglomeration of multiple databases that were previously separate, but they are on the same platform, right?
Well, yeah. Maybe I should be more specific. BreedBase is a terminology that we like to use because it's simpler and it's not focused on a crop name that would, you know, make people think that, oh, it's only for that crop and no other. But I should also say that each of these crops are actually using the same technical structure behind the database structure. The code structure is similar. So you can of course change in the code in various ways so that it fits better what the crop you're using needs, but the overall structure and code base is actually the same.
Okay, so you didn't actually go through a merge where you conglomerate the content into one single database?
No.
But does BreedBase have like a single search interface then, that because you all are based on BrAPI, or at least the similar code base, that a user could search all multiple partner databases, so to speak, from one interface?
Yeah, it's an interesting idea because so far our attention was focused on crop communities, and therefore we developed the instance so that it's geared toward one particular community. But ultimately, as you mentioned, it uses the BrAPI API, the API. So you could think of an interface that could do queries across different databases at once. Absolutely.
Okay, thanks.
Thank you. Any other question?
This is Andrew Farmer. I think we met at a meeting at Noble earlier. I was wondering, so with respect to management of genotyping data.
I'm sorry, could you speak a little louder?
My microphone is probably not very
Okay. Sorry, I'll make it. Let me. Okay, please go ahead.
Let me know if you can't hear me.
I can hear you. Okay. Yes.
Okay. I was wondering about the genotype management, and are your genotypes based on resequencing data, or is that just some genotyping assay that people are using?
We have a bit of both. It depends actually a lot on which crop we talk about. If I look at cassava, I would say that it's mostly resequencing. It's very widely resequencing, either genotyping by sequencing, or in some cases we have whole genome resequencing. We have also some low density assays such as Caspar data these days, which are more handy for the breeders. But for other crops such as SGN for cell genomics, we do have genotyping arrays that have been widely used in the community.
Okay, well, so I guess I was wondering in terms of as references evolve, you know, and genomes get better, how you guys are managing.
I missed the last part. You said as a reference genome getting better. How you? I missed the last part. Sorry.
Well, I'm wondering how you're dealing with transfer of genotyping information, new versions of references.
Right. Yeah, that's a very important question. So in the case of cassava, we experienced that a lot. We had a few changes in the reference genome over 3 or 4 years. So what we got to do was simply to realign sequencing data to the new reference, of course, and we have different genotyping protocols hosted in the database so that you can see in the database version 5, version 4, or version 6 of the data based on the different reference genome. This data is available. The database does not handle that conversion itself, but we can host different genotyping protocols. Okay. Do I answer your question?
I think so. I was also curious, are you, I think sweet potatoes are polyploid, right? They are, yeah. Okay, so how are you dealing with genotypes for polyploids?
That's a good question. My honest answer will be this. I don't work with the sweet potato group very closely, so I may not have all the answers, but within the project, they did resequence the two parental genome, ancestral genome of sweet potato. And I believe they've developed recently tools that are handling the different origins of the marker data to handle that polyploidy aspect. From the database point of view, as a database use a VCF format as a main input format for genomics, VCF can handle polyploidy fairly easily by adding an additional information field in the VCF structure. So hosting the data itself is not a tremendous difficulty. Now, I guess question would be raised how to use that data in different types of tools we may have.
Right. Yeah, I was thinking about autopolyploids and issues of SNP dosage and how those would be handled.
I know for a fact that we have a Musa-based database focused on banana resources. And we have a colleague there who has developed genomic selection using polyploid bananas, so various levels of ploidy, including diploids and triploids. The dosage data can be adjusted in those cases, as the same as you can do in potato, for example. You can score, instead of 0, 1, 2, you can score 1 to 4 dosage scores. So you can include better that variation in number of alleles within the multiple copies of your genome.
Okay, great, thank you.
You're welcome. Okay, any other question you'd like to ask?
I do, just like more about the future and which direction you plan on taking on this, because you have a great set of tools. You have a great collaboration with like phenoApps, and then you have BrAPI.
Mm-hmm.
Are you going to expand this further, or you're going to try to make a way for all these to even integrate more, or are you basically done with developing BrAPI and various other things, or is it something that's going to continue?
We are done. We don't do anything anymore. No, of course not. Yeah, so I guess having BrAPI was really the number one thing we needed to have to even envision further collaboration with other platforms. We have now achieved a level where we are able to work together with other platforms such as phenoApps, as you mentioned.
Mhm.
But that doesn't stop here. For example, up to now, we still have the need for file transfers between the apps and the database. And up to now, it was required for you to upload manually your data. We are now having some technical solution to push data from through BrAPI to push and pull data from the handheld application to the BreedBase instance. And we want to develop a lot more on those aspects so that ultimately users will not have to bother from the collection step to the storage step to bother about transferring any data themselves, but that would be handled by the application itself. So this type of application we are looking forward, but also regarding other partners, I guess there are formal partnership, but partnership can also be informal in the sense that if you use BrAPI in your own instance, you can contribute, or you can use some of the tools that have been developed by the community. Yeah.
We do have a question on the chat about, is there a article about BreedBase that you
Not yet. This is something we are working on and we want to release in this coming year. So that will be actually one of the main items for our team.
And just a question about within all this, you basically have created this really nice suite of databases that can kind of, they talk to each other. I don't know if you've looked at the Ag Biodata white paper that we put out in 2018, that we published 2018. It had a number of things about basically about how databases can talk to each other.
Mm-hmm.
And some of them were talking about persistent identifiers for data and making sure that you had a license for some of your data sharing. Is this something that you're also thinking about as you're working, making this umbrella for BreedBase for all these databases?
I haven't looked at that paper. I've seen it, but I haven't looked in all the details. I guess my question would be, by persistent identifier, do you mean an identifier that is such as what we have for germplasm, passport data, something of that nature that you can easily track across different systems? Or
yeah, one of the things that we mentioned was maybe like a DOI or even an ARC, so you can share it even outside, like if you could share this outside BreedBase.
It's not something we have necessarily implemented systematically in our system, but I can easily see that existing through, for example, BrAPI through its germplasm call, which is used to transfer germplasm information as what we call the MCPD, multi-crop passport data, and that call definitely has such type of information in there. So yes and no. It's not implemented as such, but yes, it can be easily, in some ways at least.
One of the things is that we have recommendations, for databases to try to make everything fair and shareable for people.
Yes, right.
And we, as a group, are the first people to say it isn't easy to do these recommendations. There's a number of their own databases here can't do it, or it's lack of time.
Uh-huh.
But the whole BreedBase umbrella would be almost like a great, great view to see, like, how, since you all have the same platform, how easy is it for a number of databases to have the same platform, that have a number of things, including BrAPI and phenoApp and everything that link them together. How easy is it for these databases to do some of these recommendations? It would be a great, test case, basically.
Yes. So I can talk a little bit about this. I don't think we have committed formally to what you described, but I cannot agree more with you when you say it takes a lot of curation usually when we get to, for example, integrating genomic resources that have been developed. we have the germplasm, and we want to, be as accurate as possible in terms of identification of the germplasm by providing all those codes. And really, I think, just to agree to say that because I really agree with you, it's really not necessarily a difficulty technically, but it's really a lot of curation.
Yeah.
And on both sides, from the database side, but also the partner institutions that are involved. We are lucky enough that in Cassava we have people working specifically on those aspects with us, but it's not always easy to get that implemented, unfortunately. But yes,
that seems to be like the main issue is basically time and talented people.
Resources, yeah, human resources for that, definitely.
Are there any more questions?
It was a great talk. Thank you very much.
Thank you. All right.
If no more questions, we can end a little early. Again, thank you for giving this great talk about BreedBase and all this other associated databases with it. And I'm really interested to look more into like how you did the phenoApp, kind of the circle you have of everything working together. I thought that was very interesting. So
sure.
Thanks.
I'd be happy to share the slides. I guess I can send them to you.
Yeah, just send it to me. And
all right. Okay, I'll do that.
Great. Well,
thank you so much.
Yeah, thank you.
Yes.
Thank you, everyone. Great.